Cuando hablamos de preferencias ambientales, bueno, quería hacerla en inglés, creo. Bueno, podemos tirar en inglés. So the first question that arises is why do we want to investigate environmental preferences, right? And the main reason is because the world's ecosystems are changing, right? There's a global change, um, warming oceans, modifying trophic networks, changing the pH. In aquatic systems, there's drought. There are many impacts, right, that are influencing the function of ecosystems. And core to the functioning of these ecosystems are microbial communities. So understanding how microbial communities respond to this environmental change is really, really key to forecast what's going to happen in our ecosystems under change. And then the second question that comes up is why do we want to predict instead of just measuring environmental preferences? And I took this image from a graphical abstract from a paper where they talk about this microbial dark matter. That's a concept that I mean, I don't really like the concept itself. I think it's a bit misleading, but it's this idea that the vast majority of microbial taxa remain uncultivated. And so if you think about this and you want to predict how ecosystems are going to change, and microbes are key there, most of the microbes out there are not cultured. So we need to get some sort of cultivation independent approaches that can allow us to approximate, infer, predict how these communities are going to change. And so here I'm just showing a general list of the main environmental preferences in terms of growth limiting factors for bacteria in this case. I think it's pretty general actually. And a couple of years ago, we did a perspective article where we tried to rank the degree of knowledge of these different environmental factors in terms of what we know about the growth optima of bacteria, say what we know about temperature optima, pH optima, and so on. And we saw that actually we are being optimistic here, and most of them are pretty poorly studied. And that's for a main reason. So what we saw is that the information we have comes from cultured isolates, which BacDive here is the main database that gathers all the growth information for the known strains that are out there, right? And so we were looking for information on the temperature optima, pH optima, oxygen, and salinity optima, which are just the 4 factors that are included in BacDive, factors for which BacDive offers a value, an optimum. So if you look at pH, for example, you can see the degree of bias that we have by relying on isolates. You can see that there's a very clear peak at pH 7, just neutral conditions, right? I mean, we know that the world is not buffered at pH 7 exactly. I mean, oceans are rather towards 8, and most soils are actually closer to the 6.5, 6, and so on. So there are large knowledge gaps around there. For growth temperature on the top left, you can also see that it's mostly around optimal, standard growth conditions. So pH 7, temperature 25, 30, and so on. So one can already see that there are big biases here that we should try to revert if we are really interested in the majority of diversity that is out there. You can see that the main phyla that are always represented here are the usual suspects. It's like Pseudomonadota, Bacillota, Bacteroidota, and Actinos, basically. It's the usual suspect. So we know there's much more out there, right? But it remains a pretty big knowledge gap. And so, as I said, isolation and culturing is still a very valid and needed approach to get information on microbial traits and how they respond to environmental gradients. Like you can have very detailed environmental gradients in the lab. You can modify the temperature at which you incubate things. You can add salts. You can modify the pH very finely, very precisely, and measure growth rates, right? So that's still a very valid approach. But of course, as I said, that just allows you to culture so many, just a small proportion of the diversity that is out there. And that leads to high bias. And on the other hand, if you use culture independent approaches like sequencing, we can record at least the presence and prevalence of a very broad range of taxa. Most of them cannot be cultured, but of course we have very low resolution. You at max, you know a bit where they live and what they might be doing, right? And knowing that we have these tools at hand, we conceptualize a framework where the main idea is to get the most of both worlds. So you want to capture the broadest possible diversity, but at the same time, for that broad diversity, you want to have some well-defined empirical information on their environmental preferences. So if we start from the right, you can think of a gradient. For example, it can be a pH gradient, right? Where you can record via 16S the relative abundances of different ASVs. And statistically, for many of them, you can end up calling an optimal pH, just based purely on biogeographical distributions. And for a subset of those, you might be able to retrieve representative genomes, right? And investigate those genomes, see what genes are there that might be associated with pH preference, and use those genes and that genome database, if you're able to build a large enough database, to train some sort of predictive model where you would just input a given genome from a taxon you know nothing about, but the model would look for those key genes involved in pH adaptation and then spit out some sort of prediction of the optimal pH for growth of that given taxon. That's the main idea, but of course that needs to be validated. And so the idea is to have some isolates in the lab that you can test and you can sequence their genomes. So you have information on their optimal pH and their lab conditions, and then you can also look into their genomes and verify that indeed your model is predicting the growth optima that you're measuring in the lab. So that's the main framework we're working with. And we know it works. So that's the thing that we tested a couple of years ago for pH exactly the pipeline that I just outlined for you. So we took gradients both in aquatic and terrestrial systems, like just fresh water and soil, and obtained representative genomes for taxa from which we could call a pH optimum. And then we were able to do a lot of stats on which genes might be underlying pH preference in bacteria. And we ended up fitting a model that was actually pretty good at predicting pH preferences from genomic information. So having that in mind, we revisited the list of environmental factors, and what we saw quite strikingly is that for water availability, so desiccation tolerance, drought tolerance, there's completely no information in BACDIVE or any phenotypic trait database. And that's why we planned this project, which was funded at the Plan Nacional, which is called MicroDrought Predict, and has this idea of predicting drought tolerance for any given bacterial genome from which you know nothing about. You can imagine that that can help in climate resilient agriculture, understanding how microbial communities in soil are going to respond to drought, etc. And you can have many sources of information. Some taxa might, if the trait is phylogenetically conserved, you might already know more or less the tolerance of a given taxon by just placing it on a tree. But of course, things are not so simple. So we are also interested in knowing what genes might generally underlie drought response. And what I'm going to show you in this talk, the few results I'm going to show you are related to proteome chemistry, which was something that initially we didn't think about. But then, according to some hypotheses I'm going to show you now, we think it makes sense to try to predict drought tolerance from just amino acid composition of a given predicted proteome in this case. So following literature on salt adaptation and general knowledge that halophilic taxa have very acidic proteomes, and halophilic taxa live in systems with very low water activity because it's all bound to cations, and so in general they see very little water. We thought that should also apply in the case of drought-tolerant taxa. The idea that the proteomes of drought-tolerant taxa should be enriched in acidic amino acids and impoverished in basic amino acids. And that's because acidic residues tend to capture, are negatively charged, so they capture more water molecules. And in theory, that should avoid protein aggregation when there's very little water available. So basically, if there's no water solution, proteins will start to aggregate, they will fold, misfold, and finally lose function. So we thought that to function in systems where there's very little water available, you should have very hydrated shells And that should be achieved by having lots of acidic residues and fewer basic ones. So that's a very, very general expectation that we had, and led to this main hypothesis. So we had two main objectives here. We just verify that, right? Do proteomes of bacteria from arid systems have higher prevalence of acidic amino acids and fewer basic amino acids than the proteomes of bacteria from humid systems, right? That'd be a first general question. And then the second one is, how general is this association? Is it just for a few phyla? Is it common across the tree of life? Are there certain phyla that have intrinsically evolutionary a different proportion of acidic basic amino acids than others? So for that, we wanted to gather data sets that covered either metagenomes or genomes, in this case, metagenome assembled genomes, from arid and humid areas around the world. So these are soil metagenomes, for example, on the left across Australia. Australia has a very broad aridity gradient across the continent. In the middle, you have a data set of global bacterial mags from soils. You can see taken from everywhere. And here what we are doing is overlaying the aridity raster, the global aridity raster, and just based on the coordinates, assigning them an aridity value to each of them. So in this way, you can build a data set that contains both metagenomes and genomes, each of them assigned to a specific aridity value. And here, aridity, we are taking it as some sort of proxy of exposure to drought. We have another data set that comes from actual drought manipulation experiments, but we're still analyzing the data there, so I cannot show it here. But I'm going to show you the examples of the metagenomes and the more than 2,000 mags. The main idea here is you predict open reading frames, coding regions, and from those coding regions, you can just translate to amino acids, and then do some stats to associate that composition of amino acids in these genomes or metagenomes and the aridity data that we gathered via the mapping to aridity raster and coordinates. And here are the main results. I'm just going to show you in parallel the two observations. So you can see on the top left for the metagenomic data set, I'm showing both the proportion of acidic and of basic amino acids here in the plots. And you can see that the patterns are pretty clear. Metagenomes from arid regions have a higher proportion of acidic amino acids in their proteomes and a lower proportion of basic amino acids compared to those in humid areas. So the patterns are really clear, just an increase in acidic and a decrease in basic amino acids, just as we expected. And when we look at it in a more genome resolved manner, which is the 2,000 MAG data set, we see a lot more noise because, of course, this is a global data set. So imagine we're comparing samples from Greenland to Australia, which might have even similar aridity values overall. So there's lots that can vary there. But at the same time, the patterns are holding. So we still see that taxa from arid systems have more acidic proteomes than taxa from humid systems. One can look at this in a more quantitative way, and this proportion of acidic and basic amino acids can be summarized as the isoelectric point of a proteome, or per protein isoelectric points. And you can see here that we see some very pretty sharp thresholds at very extreme aridity values, which are to the left. And here, I'm sorry I didn't mention that aridity here. The most arid conditions are those closer to zero. Here, probably it's not very intuitive, but you can see it here in the quantitative relationship. So more arid means a lower aridity index in this case. You can see that there are pretty sharp a pretty sharp change in the isoelectric point of proteomes at very low aridity. So there might be some sort of phase transition there, a critical value of aridity that triggers a very big change in the proteome level features of this bacteria. When we look at this on a per protein level, we can understand a bit more about this. So this pattern that you see here is just the proportions of proteins in any given genome or metagenome that have a given isoelectric point. So general, there's in bacteria, there's this bimodal relationship here. So there's always either many acidic proteins and there are many basic proteins. And this depends also on their cellular location and other aspects that I'm not gonna extend here. But what you see here is that indeed, for the metagenomes, for example, you can see that the metagenomes from arid regions have a higher prevalence of proteins in the very acidic isoelectric point. You can see it around 5. And they have a lower prevalence of proteins that are in the basic spectrum of the isoelectric point. And we see the same pattern more or less for the global MAC data set. So on a per protein level, we see the same patterns, not just in terms of proportions of amino acids. From that, one can derive some sort of ratio as well to see patterns more clearly. But again, it's just repeating the same patterns we've seen for amino acids. Just that in arid conditions, there are more acidic proteins, which turns in a lower ratio to the basic proportion of proteins. So it's the same pattern. And then the nice thing about the global MAC dataset is that this genome resolution allows us to investigate the pattern within phyla. And that was very key because we thought that some phyla might indeed have intrinsically, evolutionarily, for reasons that we cannot really grasp here, different isoelectric points, different prevalences of more acidic or basic amino acids. And that's actually the case. So if you look at the plot here, the second phylum on the x-axis is the Actinomycetota, you can see that they overall have more acidic proteomes. They just have a lower ratio here. So they are more acidic in general. And the same happens for other typical drought-tolerant groups like the chloroflexi, for example. But at the same time, if you look at the patterns across phyla, arid is still lower than semi-arid and humid in the large majority of these phyla, except for the Actinomycetota. For the other groups, the pattern that I showed you before is holding. So we still observe the same group, no matter the phylum you're looking at. So that hinted us at some more general evolutionary aspect here behind that might link the proteome composition of bacterial, the amino acid composition of proteomes with aridity adaptation. But finally, we were looking for some sort of validation. And what would be the best validation to remove any doubts about the effect of taxonomy than having a single genus and compare the desiccation tolerance of all the strains in that genus in the lab, and then obtain representative genomes and see if indeed the genomes from the most desiccation tolerant taxa are acidified, have higher prevalences of acidic amino acids or have lower prevalences of basic amino acids. And for that, our collaborators in America facilitated us this data that on experiments that were run there. And here's the result. So it's pretty messy data, I must say, and it's pretty tricky, and you're dealing with just a single genus, and desiccation tolerance is not also something easy to measure experimentally. But what we saw, at least for the proportion of basic amino acids, is that indeed the pattern holds. So what we see is that in more desiccation tolerant taxa, for that genus Arthropacter, we also see a lower proportion of basic amino acids compared to the ones that were more sensitive to desiccation. And the way around, we also observed, even though the pattern was not as clear, for the proportion of acidic proteins. Again, more acidic proteins in the desiccation tolerant, fewer in the sensitive taxa. And that makes us think that there is really a evolutionary aspect behind this, that amino acid composition can really be predictive of the aridity, drought tolerance of bacteria. So to conclude, we do think that it will be a good indicator of the drought, aridity, desiccation tolerance of bacteria in soil. Of course, that's a soil study. We've seen that the patterns are holding within phyla. And the most interesting thing that we're going to try now is using all this information on amino acid frequencies of proteomes to build some sort of predictive model of bacterial drought tolerance, where a model that looks for amino acid frequencies in a given proteome you know nothing about and spits out some prediction about whether it's going to tolerate or not arid conditions. If we finally go back to the original framework that we had, that predictive framework, and we feed it with the different pieces of information that we have right now, you can see that the only one missing here would be the predictive model, which is, as I said, the next step here. So we have the information from the cultivation independent approach, the data sets we gathered. We were able to assign specific genomes an aridity value. We were able to identify amino acid features that more or less correlate or are associated with aridity. And we have the validation data there, and we would try to get more isolates to have a better validation here. So the last one will be just to try to fit, to train and build some sort of predictive model. And with that, I'm just like to thank you for your attention, thank the team, the students involved, the support from collaborators, and the funding. Thank you.